All right, folks, we're going to talk about basic concepts and terminology related to genetics and genomics. It's not exhaustive. I cut a lot of information out of what was in your textbook and sort of pared it down to what I think you're going to need to know in order to make sense of later information that you will be tested on. Now, some basic concepts and terminology. Genetics is the study of inherited traits. A lot of it is under the umbrella of Mendelian genetics, but there are other subspecialties. What is genomics and why is that different? Genomics is the study of the function of all of the nucleotide sequences within the entire genome of a species. Every species has its own genome. Every person within the species has subtle variations. It includes study of the genes in the coding and non-coding regions of DNA, interactions between and among genes. We have genes that tell the cell when to divide and when to stop dividing. We have genes that tell the cell when to self-destruct. We have genes that determine the pace of cell division and regeneration. And then the interactions between the genome and the environment, which more accurately falls under epigenomics. But genomics is a much broader thing. The terms are used interchangeably, but the difference is that genomics is a broader science that includes genetics. That's the test question, so just so you know. Okay, basic concepts and terminology. And I have a little graphic here as I go through. We have a gene. A gene is like one of these little areas of DNA. It's a specific segment of DNA that contains instructions for making amino acids that combine into specific proteins. All of the enzymes, all of the structures in your cell, things that regulate the body's functions are dictated by genes. And then we have DNA. DNA is the raw material inside the cell that contains the genes. Think of it as a ball of yarn, and a gene is like a section that you knitted into something that makes sense. The human genome is the entire sequence of genes for our species. Within that human genome, there is individual variation, but there is, a place on every chromosome that performs a specific function within that genome. It has to be in the right order. A chromosome is a little package of DNA that houses specific genes. So there's your chromosome. In humans, there are 23 pairs of chromosomes in a normal cell. Now, there are exceptions. When we talk about ploidy, we'll talk about those. But in a normal human cell, there are 23 pairs of chromosomes, total of 46. They are located inside the nucleus of the cell for the most part. There are a few pieces of DNA in the mitochondria, the powerhouse of the cell. They are all inherited from the mother. There are no paternal, or at least unless the science has evolved more rapidly than I have. The mitochondrial DNA is all maternal because it came with the ovum that was fertilized. And most of them are in the nucleus. And most of what we're interested in is the nuclear DNA. Okay, basic concepts about genes, chromosomes, and DNA. Most of the DNA is found in the nucleus of the cell. Hint, hint, that's important to you. Small amounts found in the mitochondria. Every nucleated cell in the body, red blood cells don't have nuclei, but everything else mostly does, contains the entire genome. Even though it only expresses a few traits that are very specific to its type of cell or tissue, it has the whole script for the entire production. The only ones that don't are the mature sex cells, spermatozoa and ovum, and they contain half of an individual's material, genetic material. Somatic cells, every cell that's not a sex cell, are called diploid. They have 2 copies of each chromosome, one from mom and one from dad. There we go. Sex cells are called haploid. They have half the individual's genetic material. It's still half from mom and half from dad, but there will only be 1 chromosome at the end of meiosis. The human genome contains about 25,000 genes, more than most insects or, other organisms, but less than a lot of plants. Go figure. Cells have the capacity to switch genes on or off to ensure that each cell performs a unique function. A cell that is part of the nerve of your foot is always going to act like that kind of cell. It's never going to act like a bone cell or a cardiac cell or, cardiac muscle cell. It's always going to act like a neuron in your foot. So to make an analogy, think about a big epic motion picture, something like Avengers Endgame. You have the entire script, and everyone in the cast, except for Tom Holland, because he gives out spoilers, gets the whole script. But they only read their own lines, because a director tells them which lines they need to read. Now that's done through histone modification, not for today's content, but just know that every cell in your body has all the information it takes to make a human, a whole human. It starts out as undifferentiated stem cells, but once they're cast in a role, they only read their own lines. Sex cells get just the information they need. That's why Tom Holland only gets his own lines. He has about half the script. Probably less, but whatever. A sex cell gets half. Chromosomes, a little more about chromosomes. So here is a picture of a chromosome. They organize and preserve the structure of DNA. Think of that ball of yarn. Yarn is just the raw material. If you just let it get tangled up and knotted, you can't knit anything with it, right? So it has to be preserved in some way. That's done by packaging genetic material into a temporary structure called the chromosome, which lives in the nucleus of the cell. You can speed this video up, by the way. I won't prevent you from doing that in Edpuzzle. But each chromosome has 2 chromatids. They are, watch my pointer. Can I get a pointer? Hold on. Shoot. Turn on the laser pointer. There we go. Here is the left chromatid, and here is the right chromatid. These are bound together at the center by a structure called the centromere. Okay? The short segments at the top are called P-arms. P, because it comes before Q, therefore that should be shorter. And the longer arms, which are called Q-arms. Every chromosome in that karyotype, I'll show you a picture of a karyotype at the end of this presentation. Every chromosome has a different sort of a structure. We go from chromosome 1 is the biggest one, the last chromosome, the sex chromosomes are at the end, but they get smaller as you go down. So it starts with the biggest chromosomes, ends with the smallest ones, and that's how we display those. They are capped on the ends with telomeres. It's like the aglet on a shoelace. Aglet is that little thing that keeps the shoelace from unraveling. And if you've ever lost an aglet, you know that everything gets frayed and lost. So they kind of preserve the integrity of chromosome. Now, DNA is incredibly complex. It must reproduce during normal cell division. So it has to sort of rip itself in half. And it does so. I'll talk about hydrogen bonds in a minute. And reproduce itself exactly, perfectly the same sequence, the same places. So DNA in each new cell has to be an exact replica of the parent. Now the problem is if you stretched out all the DNA in one cell, just one, think about how small a cell is, think about how small the nucleus is, think about how microscopically tiny the DNA within that nucleus is. But if you stretched it out from one end to the other, it would be 6 feet long. That is a lot of raw material. To prevent damage, there are some proteins that the body makes called histones. They bond together and they form a little spool to wind things up. And so you see here we have 2 chromatids, right? Left here and right here. They're bound together by the telomere, that little ribbon, so that we don't end up with this. It keeps the genetic material organized in its exact sequence. It unfolds when it needs to. There are histones that also bond to genes and switch off aspects of it. And we'll learn more about that in epigenetics. But this is all a very incredibly intricate process. Now on the chromosomes, genes have an address. It's called the gene locus. And every chromosome is numbered, biggest to smallest, like I said. This picture is chromosome 11. And through the Human Genome Project, we found that the address for the genes that produce insulin are on chromosome 11, the Q arm, which is the longer bottom half, in this little region that they call number 13. So if you wanted to send a postcard to the genes that produce insulin, you'd have to send it to 11q13. And that becomes important to you guys later, because as we can sequence an individual's genome, we can look for errors. And that research guides how medicine develops. But anyway, all right, DNA is composed of 4 nucleic acids that are known as bases. I'm not going to worry too much about purines and pyrimidines. We're just going to talk about adenine, thymine, guanine, and cytosine. They are the building blocks of DNA, and they fit together in a certain way. Adenine and thymine go together. Guanine and cytosine go together. It's sort of like having 2 different size Legos. They fit. Each base attaches to a sugar called deoxyribose and a phosphate, and they form like a string that holds the beads together in a necklace. You do need to know the bases. The other stuff is sort of helpful to help you understand, but you do need to understand the bases. So base pairs consist of 2 complementary nucleic acids held together by weak hydrogen bonds. Now remember, in chemistry, probably distant memory for you guys now, but there were covalent bonds. Those bonds are very hard to break. Hydrogen bonds are ionic bonds. They are very easy to break, and that's important because when you replicate DNA, it has to split right down the middle, right down those hydrogen bonds. And then it can combine with the new copy of the other strand. Adenine and thymine each have 2 sites for hydrogen bonds. They're a complementary pair. They always go together. Guanine and cytosine each have 3 sites for hydrogen bonds. They're like the Lego Duplos that don't fit the regular Lego. So they always go together. The strands separate during cell division. You can predict the sequence of bases on one strand of DNA if you know the sequence of bases on the other strand. This becomes very helpful when we discuss disorders where it's a mutation that causes a change in the base. If you know what you're supposed to have and you're proofreading to see why this person has this disease, it kind of makes sense. One thing that you are going to need to be able to do is to predict the sequence of bases on a strand of DNA by looking at the sequence of bases on the opposite strand. And we are going to let off here and come back with some more fun.